Hi Year 12s, this is our third video in PowerPoint about DNA profiling. Now in the last two videos I've said there's 4 videos, that's a little bit of a lie, there's actually 6 videos this week. In this video though we're going to be looking at gel electrophoresis. Now as well as actually looking at gel electrophoresis, we're actually going to be doing an experiment where we actually physically have our own gel electrophoresis in the lab this week. Not to the same level as they'd be using it in an actual research laboratory, but you'll get the idea of how we can physically set up gel electrophoresis in a lab. So, first off, what exactly is gel electrophoresis? Well, basically it's a process allowing us to separate DNA molecules of different sizes. So what happens is you have basically a little tray of agar gel, okay, and it'll have little holes called wells, and in each well you will put some samples of DNA. Now the actual agarose gel itself is connected to 2 electrodes, and a battery will consistently make sure that there's an electrical potential difference, so there's 2 different charges across that electrophoresis tank. Okay. Now how this actually works is that DNA is negatively charged, and that's because of the phosphate groups in the polynucleotide strands, so they will be drawn towards the positive electrode at the other end of the electrophoresis tray. Okay. As they move towards that positive side, the DNA fragments will be physically separated based on their size, because based on their size, they will move at different paces through the gel. So the larger the molecule of DNA, the slower it will move through the gel, and it won't get as far as the process continues. The smaller the particle or the molecule of DNA, the faster it'll go through the gel, and it'll get further through gel electrophoresis. Now, it's not enough to physically just have the unknown samples of DNA in the agarose gel. You also need to have a separate marker DNA that you know the sizes of the fragments in that set of DNA. That way you can physically compare the unknown samples to those known samples and actually determine the size of the unknown samples of DNA. So this is a little bit what it'll look like. Ours won't look quite like this. We're using plastic trays and alfoil. Same battery pack type situation. You've got your positive and negative electrodes. Okay, now because DNA is negatively charged, when this turns on, they will start to move through the gel towards the positive electrode. It's negative is attracted to positive. Okay. As I said before, the smaller the particles, the faster they'll move, so they might end up down here. The larger molecules are slower, so they might end up at different points depending on their size, but they won't end up as far as the smaller ones. We also will physically add fluorescent dye. Now, we won't do this part in the lab. We're actually not even going to be using DNA. We're going to be using food coloring molecules, so you can tell the different particles that way. Okay, but in the actual laboratory setting, they'll be using fluorescent dye, which will cause the DNA molecules to fluoresce under UV radiation. So once they physically get this gel electrophoresis done, they'll put the entire sample underneath some UV light so they can create that DNA profile so we can analyze the fragments.